Welcome to your Anatomy Online virtual experience. Today we're going to be looking at a virtual microscope. Since you don't actually have a real microscope, like I demonstrated in the video the other day, we have here a virtual microscope. And this is courtesy of the North Carolina Biological Network, and you'll get to this microscope by using the link that was included in the announcement about microscope use, and you can also see here at the top of the screen the link for that as well, or the address. So here we have this virtual lab environment here, and down here at the bottom we have some options. We have the guide, and if we click on that, it's basically some things we can read about a microscope here. We can go through a general overview of a microscope, the different pieces and parts, those things that I did in the previous video, and probably to a much greater degree of detail here. But if you would like a little bit of a reminder about how the microscope works, what the pieces and parts are, and all that good stuff, you can check that out at the Reference tab, or the Guide tab. Then in the Learn tab, if you click on that, it will take you to, in this case, a lab environment, and each of these little question marks is actually clickable, and then it pops up a description of what that particular thing was. So that's chem wipes. This particular little box over here is the box of slides that we'll look at here in a little bit. The coarse adjustment knob, or coarse focus, fine focus, base. Those things should sound familiar to you. So that's just a reminder of what each part looks like and where it's at. And some other handy little tools we would use in a microscope lab, like lens paper, which we don't have to do for a virtual scope, but a real scope, lens paper is always a good thing to have. And immersion oil, we wouldn't use, but in your medical microbiology experience, you'll get to play with that. I think I talked about that in the video the other day as well. In the Explore tab here is really where you're going to use the microscope. So you notice here that the only thing with a clickable question on it is our slide box. So if you click on that, it opens up a list of different slides that you can look at. The first one is sample slides, and for the sample slide you can start out with this if you want. When you click on a particular slide, it loads a view like this. So this is what you might see here in this round area if you had your microscope on 4x. Remember, that would give you a total magnification of 40. And if you know that this is a letter E, you can't really see it clearly here. And down here at the bottom is a coarse focus, a fine focus, and a light adjust. So instead of actually turning the dials on the microscope, you're just going to move these little slider bars back and forth. And remember, we always start with the coarse focus. So we're on our lowest power, the 4x. And so we're going to take our coarse focus and we're going to start turning it. And notice the microscope over on the left, the little virtual microscope there. As we move the coarse focus, the stage goes up and down, just like I told you it would in the video last time. And in the circle view here, we're seeing what was very fuzzy looking something. The more we adjust that coarse focus, the clearer it gets. So we get to about right here, and if we go farther, we'll see that it starts going the wrong way. So we'll back it up until it looks like that's pretty close. Then we can use the fine focus knob here, and we can adjust it a little bit further, and it looks like about right there is as good as we're going to get. So the coarse focus got us pretty close. The fine focus got us a little bit better. If we come over here to the light adjustment, and let's say we make it darker just for the fun of it, and on this particular specimen of the letter E, not much happened. But if we go up here to the brighter end of the scale, look what starts happening to that E. It starts to fade away. So we're getting too much light. So again, too much light can actually glare out the object and interfere with your view. So on every slide, you'll want to adjust the light until it looks about as good as you can get. If you wanted to go to the next level of magnification here at the 10x, we would just click on that, and you'll notice over here on the side our objectives. As we click on a different objective power over here, the virtual microscope does in its view what it would do if you were in a real scope. So on 10x, there's the view we would get. So when we go up to a higher power, we have the opportunity to adjust our focus a little bit better. In this particular case, I don't think we'll probably get much better either way, because a letter E is a pretty big thing. But now we don't see the whole thing. So when you're zoomed in a little bit, you actually have the option to go up here in the circle and click and then drag that circle, that specimen around, so we can see the different parts of it if you'd like. And on a letter E, that's not very exciting, but you get the idea there. Let's say we wanted to go to 40. We can click on the 40. So now we have a total magnification of how much? 40 times 10 would be 400. And again, our focus probably isn't going to get much better on this because it was just a letter E. And we could go all the way up to 1,000 total magnification here on the virtual scope, which we would not be able to do on a regular scope in the lab because we don't use this. But if you wanted to, so if you want to go look at things in the slide box here that doesn't necessarily pertain to our course, when you get to that 100x objective, it says you need to add immersion oil. So we would go over here and click on our little immersion oil, and it would virtually add a drop of immersion oil, which would allow that objective to work. Now in this case, it's zoomed in so much it looks like the screen's totally black. That's because we're really zoomed in. But if we drag the image around, we would be able to see that it's all there just like it was before. We're just really zoomed in on it. And if we go back out to the 4, it says we shouldn't do that. So what you really want to do is you want to, just like in real life, is you would go from 100, okay, we're going to say done, back to 40, and that's okay because you're backing up. So reversing your process when you're finished is always a good thing. And then it says, well, you've used immersion oil. So you might want to get some lens paper and clean your slide, clean the objective off. So if we clicked on the chem wipes, that's not a good idea because that contains chemicals that can be harmful. So let's try our lens paper over here. It cleans it. Everything's nice and good and ready to go. So then you could work your way back down to our lowest power if you wanted to. Then we would have what we saw originally as the letter E, and it was a little bit off, so we drag it back over and there we go. If you were done looking at that, you would click the remove slide option. So what we're going to do in this class is not so much look at the sample slide, although it was interesting. What we're interested in primarily are the human's tissues. So down here on the human tab, we would click that, and it gives us a nice little list. Adipose, areolar, blood, compact bone, elastic cartilage. There's a decent little list of tissues here. And not necessarily everything that we would use in a regular lab setting, but this is a pretty good selection. And it's all preloaded in here. So let's say we wanted to look at bone, let's say. You would select the bone slide and do the same thing we did with the E. 4x objective here, come down to the coarse focus, adjust until it looks like it's pretty good. That looks pretty close there. Fine focus, see if we can make it better. Looks like we can make it a little better. And here's going to be a more dramatic example of the light adjustment. If we lower the light adjustment here, it actually made some more detail pop out. But if we have the light too high, very quickly everything starts glaring out. So on each one of these, you'll want to adjust the light intensity until you feel like you've got the clearest image, and then work your way up to whatever power you need to see things. So for the bone, we can go in quite a bit further here and still get a nice view, I think. Okay, so there's one of those little osteons, a functional unit of bone tissue. And remember, when we go from one power to the next, tweak your focus a little. So That looks pretty good about right there probably. And then we might tweak the light a little bit as well. And what you might often find is the darker you make it within reason, the more of the detail pops out that was always there, but the light perhaps was just a little bit too bright. If we go to 40, we're probably going to be zoomed in so far on this one. Yeah, we can see details, but we can't see the whole little unit there, so we lose appreciation. So each type of slide we look at will have a different level of magnification that works the best. And when I give you an assignment on this virtual microscope, what you'll do is you'll come in and you'll select the slides that I've told you to select. You will focus through these powers until you get to the power I told you to draw it on, and then you'll draw a picture. So right now, if you were going to turn this in, you would draw what you see here on the screen in the little circle. So you would draw this particular ostion right here, and you don't have to draw all of them, just one of something will do the trick probably. So draw that one ostion there to the best of your artistic abilities. You will label the picture, and I'll give you guidelines for how to label your drawings, and then you'll just take a picture of those drawings and email it to me. Now, if we were in class, you would do those drawings and turn them in, and I would record them and hand them back to you, so you'll just electronically submit your drawings, and away we'll go with that. I'll get your grade in the grade book, and life will go on wonderfully. So that's kind of the process to use this virtual scope. The learning curve's probably a little shorter because you're not as intimidated by this scope than you would be by a real thing in front of you in a classroom in front of other students. But hopefully this little tutorial here gives you an idea of sort of what we're looking for there. If we go back to the main screen, we'll get our options of, you could go over here to test and other options and play around with some other things. But primarily what we're going to do is use the Explore tab and then look at those human cells. And I'll give you examples, I'll give you assignments as we go through the semester from that list of slides. After we talk about those tissues, then you'll look at them on the slide, draw a picture of it and email it to me. And so that's going to be part of our virtual lab experience. Hopefully this tutorial was useful and helpful, and I'll see you next time.